Okay, hello everyone. Welcome to another episode of Science and Technology Q&A for kids and others. And I'm happy to try and answer questions about all kinds of topics about science and technology, both things you think are elementary and things you might think are complicated and advanced. I'll see what I can do and try and explain them as best I can. Let me see, we have a number of questions that got saved up from previous times. Oh yeah, here's an interesting one from Hamid. What is the physical limitation of the growth speed of humans? Can you get from 10 kilometers to 90 kilometers in a month, theoretically? Okay, that's an interesting question. So, there's a whole issue of how things grow, what actually is happening. The fundamental way that ultimately organisms grow is by making new cells, by having one cell divide into two. And in the case of us humans, we've divided, well, our cells divide up to about 50 times. And so we keep on doubling the number of cells, although it's more controlled than that when we build different parts of ourselves, so to speak. But fundamentally, what you have to do to grow is to create new cells. And in terms of when people put on weight and so on, it's not all just creating new cells. It can also be kind of having more water retained in existing cells and things like that. But if you really want to grow, you have to make new cells. So one question is, how quickly can you do that? What's involved in making a new cell? Well, you have to start off with the DNA, which is the specification of what proteins should exist in the cell and so on. For us humans, DNA is 6 billion base pairs long and separated into 23 chromosomes. And what happens if you want to make a copy of a cell, the first thing you've got to do is to copy that genetic information. And that process, the way that works is the DNA, which has two strands in it, the strands pull apart, and gradually one strand will refill with a complementary other strand. And that takes a certain amount of time to happen. Then other things have to happen in the cells. You have to copy the DNA. Then the various different other pieces of the cell have to also get replicated, like our mitochondria that produce energy originally started as quite separate organisms, but they've been sort of living inside us for the last few billion years. And they have to, have their own replication. And then the actual surface of the cell has to split apart and create a new cell. So for different kinds of organisms, and it depends a bit on the length of the DNA, it can take different amounts of time for cells to replicate. I don't actually know what the time for cell replication is for humans. I'm guessing it's on the order of 6 hours to a day, something like that. For some bacteria and other simpler cells, it can be a matter of 10 minutes or something like this. But I think for us humans, the time for the actual process of replicating cells, there's a certain limit to how quickly you can grow just on the basis of replicating cells. Now, there's also the question of, well, if we grow, which part we have to grow in the right proportions? I mean, it can't be the case that, there's some very early organisms back in, I don't know, the Precambrian period, from these stromatolites, for example, which are some early organisms that you find distributed and you find fossils of. And I think they had the feature that they just grow and they make sort of blobs and they keep on growing. And they don't really have any kind of structure or control of, oh, I want to grow something where I have an arm and it's a certain length and things like this. We have much more detailed control of growth. I mean, it's the thing to remember is, of the two biggest kingdoms of organisms, the plants and the animals, have different schemes for growth, because plants are particularly distinguished by having rigid cell walls. They have cellulose cell walls. And that means that if you're growing and you're a plant, whatever's grown so far has to be left the same. And if you're going to grow an extra branch, that's kind of a pure add-on. You don't get to kind of deform the thing that's already grown. So plants have this strategy, particularly the branching strategy. Sort of everything about plants is you grow a thing and then you grow branches to that thing, whether it's roots, whether it's branches above ground, whether it's features of leaves, all those kinds of things. It's all about you lay down something and then you add something on. For us animals, it's a little different because our cell walls are deformable. And so we can expect to have things where, oh, cells grow and it just puts more pressure and these other cells are kind of squashed up in some way, and gradually more cells get formed and so on. And you can have more diversity of growth because you have this possibility. You don't have to go, it doesn't have to be rigidly growable at every stage. And we have all sorts of different strategies for growth. But so, for example, one of the things that happens for us is we have skeletons, and it's always and that they form the sort of framework on which, you know, the flesh is put on the bones, so to speak. The bones define the overall structure, and then one's kind of hanging the muscles and everything else on those bones. So there's a question: how do the bones grow? And the typical thing is that there are growth plates. So, for example, our long bones, like in our legs and so on, they have growth plates at the ends. And those are the places where cell division is happening, and you're adding more to the bones. So the typical kind of scheme is you've got this long bone, and it just keeps getting longer and longer and longer. There's always a mystery of things like the skull, which is something which has to be sort of a closed container, and it has to stay a closed container. How does it manage to grow? And the answer is it has these separate plates and it has these suture lines between the plates and things. It grows at those suture lines, and gradually, by the time you're like 40 years old or something, everything has solidified. And there's no. But if you're for very young kids, you can still there's still lots of stuff that's soft and so on, and it's still that there's some. I mean, that's how it succeeds in growing, even though in the end it's a fully encased thing. But in any case, so for humans, there are these sort of particular schemes of growth, particular ways that things grow. Like for example, when we grow out capillaries in our bloodstream, small blood vessels, the fundamental strategy there is actually very much like what plants use. That is, you grow a big blood vessel and you grow little branches to that, and then branches to that, and so on. And eventually what we have to do in order to supply oxygen to all our tissues is we have to have branched and branched and branched enough times that we're within 2/millionths of a meter or something of every place in our body. There has to be some little thing that's supplying blood to supply oxygen to make those tissues work. But so that's another scheme for growth. And another one's in our lungs, the little what are they called? The bronchioles and then the alveoli. The whole tree of our lungs are like a plant tree. They branch about 23 times, I think, branching progressively smaller and smaller and smaller sub branches, and they grow out with big branch, then that sprouts smaller branches, and it keeps going a series of branching times, and different things grow in different ways. I mean, we have, you know, I think a trick that's used before we're born is, you know, we start off with kind of just sort of flipper paddle hands, and then the cells between the fingers die off, and that's how we end up with these separated fingers. And that's another sort of trick for growth. But when you ask how quickly can humans grow, I think the limiting factor will be the speed of cell division, and probably the DNA replication is probably the limiting step for that. And I think that will determine the maximum rate that cells can grow that way. I suspect that you can get sort of a certain amount of kind of rapid increase of mass just by having more water picked up by cells and so on. And that happens, the way to understand that, when you have a cell, it's got this kind of gelatinous stuff inside it that's mostly water. And there's sort of a question of how much water, how much of different kinds of things get picked up into the cell. And that's a whole story of this thing called osmotic pressure. So the idea is, if you've got a cell, and let's see if I can get this right quickly. If you have a cell and it's basically got a and it has, okay, so there's and it's got this sort of water with something dissolved in it inside, and then outside is just plain water. There's a certain tendency for the things that are dissolved in the water inside, there's some probability they'll escape to the outside, and gradually it will equalize the amount of dissolved stuff. cells fight against that by having active transport of materials into and out of the cell through pores in the cell membrane. So a cell membrane around the outside of a cell, it's mostly this thing called the lipid bilayer, and that consists of particular molecules, that sort of cylindrical shaped molecules roughly, that align themselves so that they have one end inside the cell, one end outside. And I guess that there are two pieces of that. But in the end, the sort of surface of a cell is mostly this kind of almost crystalline type thing that is this arrangement of particular lipid molecules that are long molecules. But in the surface of the cell, in addition to this basic sort of bag around the cell, there are a large number of specific pores that kind of are drilled through the surface of the cell that do things like transport materials in and out of the cell, and also do things like, for example, there are things that will expose pieces of RNA that were in well, they'll expose basically they're exposing actually pieces of protein that are let's see, how does this work? This is the part of the way the immune system works is it says, what are all the things going on inside the cell? Let's expose little broken down pieces of that on the cell surface so that the immune system can come and check that this cell is okay, so to speak. And that's yet another thing that's transported from inside the cell to the cell surface. But there's also lots of pores in the cell surface that transport, for example, potassium and sodium into and out of the cell. And that's something where there's active control of those, of the amount of those things in the cell. And the way, what are those pores? Those pores are proteins. And in particular, so a protein is made from, it's specified from DNA. And but a protein consists of this sequence of units that are each an amino acid, about 20 amino acids. And so from the DNA, one's specifying, okay, this protein should be I don't know, almost all the letters of the alphabet are used to specify amino acids. So I don't even have to know my amino acids to be able to say this is a B, an X, a this or that or the other, all in a sequence in this protein. And the backbone of the protein is always just a sequence of amino acids. But when a protein, the protein, once you've made that kind of backbone, the protein then just sort of folds up. And so, for example, there will be pieces of the protein where there are pieces of the sort of sequence of amino acids where those things always like to be on the inside. And there are other parts where it always likes to be on the outside. And different proteins make very different shapes, like actin, the protein that's in our muscles, is a long, it's a triple helix, I think. It's a long, stringy protein. Or something like hemoglobin is a protein that just has a little cage structure enough to fit one iron atom to transport oxygen in our blood. But different kinds of proteins have different shapes. And there are two characteristic pieces to protein shapes, things called alpha helices and beta sheets. An alpha helix is a sequence of amino acids that will tend to make a kind of tube structure. And it's those alpha helices that are used by the transmembrane proteins that transport things like potassium and sodium and so on into and out of cells. They make little tubes, and those tubes are controlled by other parts of the protein. And that's kind of how you maintain active, you actively maintain the concentration of things inside the cell. When cells replicate, as I say, what happens is the lipid bilayer membrane, I'm trying to remember exactly how this works, how you go from one closed sphere of cell to another one. I mean, when you see things like single-celled organisms, they tend to sort of pinch off and then the two pieces break apart. I think that's how human cells do it as well. Not absolutely certain. And there's a question of sort of how that happens. How does it even control the shape of the cell? The answer is there are little strings, effectively, that are pulling, that are deforming the shape of things, and that's and those are operating probably. Actually, don't know exactly how that works. My guess is that it works by the same kind of methods that a bunch of these things work by, which is literally a little ratchet type thing where a one component is literally sort of walking one ratchet step after another to move this string of protein around. But anyway, so So I'm guessing that there's an upper limit on how fast we can grow. There's some organisms that grow pretty quickly. I mean, some plants can grow pretty quickly, and so on. One thing to mention about humans. So one thing that determines our growth rate, I mean, it's a pretty complicated system, but human growth hormone secreted by the pituitary gland inside our brains. The pituitary gland is a gland that just happens to be situated in the brain, but it's mostly secreting chemicals that are hormones. Hormones are kind of the global signaling chemicals used by biology to determine sort of what happens in the system. One of those hormones is human growth hormone, and that is the thing that kind of signals this process of at least for humans after they're born, signals the process of, okay, add some stuff. And one of the things that's really weird is that the pituitary gland doesn't just release hormones continuously, sort of all the time. It tends to release them in sort of I'm not sure if it's actually in pulses or whether it's more at certain times of day. But so that means that this, what you can actually see, I think people have measured this in kids when it's a question of how do your legs grow and so on. They actually grow a discrete amount, like every night they'll grow a certain amount. And that's induced by secretion of human growth hormone, which then produces a cascade of chemical processes that lead to cell division, et cetera, et cetera, et cetera. So it's kind of a weird thing that you would think growth was always a very continuous thing, but actually because of the control from human growth hormone, it can be in little jumps, so to speak. Anyway, that would be my thought about that. Okay, another biology question here. You are really challenging my knowledge of some of these things. A question from Grace, about the function of the human heart. Is it best understood as a pump or in a different way? Well, ultimately, yes, it succeeds in pumping blood around the body. The way that it is a strange and very clever object that has it's made of muscle, and a kind of a different kind of muscle than the muscle that is part of our sort of conscious control parts of our muscles that we think to have to move. Heart muscle it's a muscle. It has a pretty complicated morphological structure. It has a pretty complicated structure of valves and this and that. The pulses that the heart produces there are nerves, venal nerves, right? I think that are sort of that are part of the non-conscious control of our brain. They're the sort of very fundamental things in our brain that produce electrical signals that kind of stimulate the heart muscle and cause the heart to contract. Now, once you've sort of put in that electrical signal, the structure of the heart muscle is such that you have this kind of wave of contraction that goes around the heart that causes it to beat in a certain way. And that's good when it works that way. If you interrupt the way that the heart is beating by giving it some other electrical signal, like if you do the thing that you absolutely shouldn't do of one hand on one on a plus electrical outlet, another a minus, so you pass a current across your heart. Very bad thing to do. That's why for people like me who are sort of if ever I do any kind of electrical thing, I'm always like, put one hand in your pocket and use only one hand and make that do, because that way at least you don't get an electric current that goes across your heart. But in any case, the thing that happens if you can get if there's sort of other electrical signals is that you can confuse the kind of usual contraction sequence of the heart, and it goes into this process called fibrillation, where it's kind of just sort of randomly contracting rather than doing its systematic contraction that it does to pump blood. And that's a bad thing. And that's why there are these defibrillators, which are intended to give shocks to the heart that break it out of this process of fibrillation. Roughly what happens is, and this has been understood over the last, I don't know, 30 years or so, that in the heart has, the way it's constructed, it has a sort of fundamental liking for beating at for periodically beating, just contract, expand, contract, expand. That's what it's sort of, that's the attractor of what it wants to do. And if you just sort of poke it in more or less the right way, that's what it will naturally do, contract, expand, and so on, in the way that it's supposed to operate correctly. But what happens is, if you kind of kick it out of that domain where it's doing this kind of contract, expand thing, you can get it into a different domain where it's not on that attractor. It's kind of like, what's a good analogy of this? Let's imagine that we have some sort of ball going around in a track, and normally it will just sort of go back. You know, if you kick the ball out of the track, it will normally just sort of roll back down the hill and go around the track again. But if you kick the ball far enough away, well, maybe it'll go into that other area in the middle where it's sort of randomly moving around. And for the heart, there is this so-called strange attractor, this other possible mode of motion, which is not effective in pumping blood around, and that's this fibrillation process. And the usual sort of the theory of defibrillators is you give another electrical shock to the heart, that if the fibrillation started with an electric shock, or if it started some other way, you're just giving it an electric shock to sort of kick it back into this attractor, this periodic attractor that's its correct expand, contract kind of motion. So at least that's you know, one thing it's worth thinking about in the history of science. When people were kind of wondering in ancient Greek times and so on, how did humans work? And you know, one thing that's really obvious about us is we think, and then people were saying, well, where's the thinking happening? What's the machine? What's the mechanism for thinking? And they look at humans, and it's like, well, there's a lot of stuff that's just sitting there, and then there's the heart. And just like as we think, you know, we think about things and things change and things are happening all the time. Well, the heart is something where we can, you know, tell that it's beating. And so there was sort of an immediate thing, oh, well, if that's the thing where there's change happening, and thinking is a place where there's sort of change happening, those two must be related. And so that was kind of a scientific theory for a long time, that, you know, the heart was something to do with thinking. And it was only much later that it became clear that, you know, brains were really the center of that. And it's something where it's sometimes it's hard to identify, you know, when you look at some piece of us biologically and you say, what's the function of that piece? You know, I'm always amused by the fact that when I learned biology when I was a kid, there were these parts of biological cells where it was like the Golgi apparatus was, you know, in the textbook that I had, it said it's a part of a cell of unknown function. Well, of course, now we know it's all involved with protein synthesis and so on. But there are these sort of features of biology where people say we don't know what it's for. And sometimes that produces tremendous confusion and even sometimes terribly crazy medical conclusions, because it's like, well, you can take that part out and it'll all be okay. But just because we don't know what it's for doesn't mean it's really not for any purpose. You know, there are many places where in the course of biological evolution, some things are the way they are because that's the way they were. You know, we share a lot of detailed biochemical features and so on with organisms that existed at least 1 billion, maybe 2 billion years ago. And they've been sort of grandfathered forward for, what is it, now must be, you know, many billions of generations, so to speak, since those species, and we've still got the same stuff. And some of those things are like, well, that wasn't a very good idea, but it works well enough that we can still keep going with it. And if you say, well, why is it that way? The answer is it is that way because that's the way it got chosen sort of randomly 1 billion years ago, and we've still got the results of that. A famous example of that is in our eyes. The eye of the type that we have has been sort of invented in evolution maybe 3, 4, 5 times. But in our invention, and like the king crabs and things like that have eyes like ours, but they came from a different evolutionary path. But for us, one of the things that's sort of a piece of bad design. As light goes into the eye, eventually it's going to hit cells on the retina, which are going to turn it into electrical signals. But a bunch of the cross connections about retinal cells are in front of the retina. So it's like, that's bad design. If you're going to have better optical properties, one would think you have those cross connections behind the cells that are taking in the light. But presumably it's bad design. Maybe it's, quotes, designed that way through the course of biological evolution. It's been selected to be that way because there's actually a good reason to do it that way, but we just don't know what it is yet. But there are many of those types of things. All right, let's see. Oh boy, another medical type question. Humans exposed to prolonged microgravity suffer from well-known health issues. Short of 1g of gravity, do we know the minimum gravity required to prevent such issues? I don't think we know that. How would we know that? So, first point to make, on the Earth, there's gravity, things fall, the amount of gravity on the Earth is measured by the acceleration due to gravity, about 9.8 meters per second per second. If we are on the moon, for example, the gravity is 1/6 the gravity on the Earth. If we're on Mars, it is, I don't know, it's somewhere between, maybe it's 50%, 60%, I'm not sure. It's something a bit smaller than the Earth. If we're in space, in orbit, for example, and we're not firing any rocket engines or anything, we will be weightless because kind of everything is falling at the same rate. And that means that we effectively have zero gravity. Now, so when astronauts go and spend, 6 months, a year, whatever, on the space station, they are in microgravity most of that time. And it's called microgravity because there is usually, because of small acceleration forces associated with things like the fact that the Earth is not a perfect sphere and things to do with the slowing down, speeding up, those kinds of things. There's but for all practical purposes, it's zero gravity. And so that means, things don't fall any particular direction. You put something, you put a blob of water in the middle of the space station, it'll just sit there and make a big spherical blob typically. It won't fall in any particular direction. So the question is, what happens to us humans under those circumstances? And the answer is, it's not totally good, because there are all kinds of things like our bones and so on. We maintain our muscle strength because we're actually walking around in gravity. We maintain kind of the mineralization of our bones because there's actually pressure on our bones and things like this. All these kinds of things, we were built by evolution, in a sense, to operate in 1g gravity on the surface of the Earth. And so anytime we go into a different kind of gravity situation, it's, we weren't designed for that. It's outside of the design parameters of the human, so to speak. So typically astronauts will do all kinds of exercises to try and maintain muscle mass and bone density and things like this. How would you make, if you were going to send people off for a really long time in space where they're weightless, what you can do is you can sort of create artificial gravity by having a centrifuge. So, for example, if you for example, take some I don't know, a glass of water or something, not a glass, some other thing containing water, and you put it on a string and you kind of whirl it around, you may know, a bucket, that's a better thing to do it with, has a handle as well. And you kind of whirl it around, what you'll find is that the water in the bucket, even if it's, so long as it's going round at a certain speed, it will be the water will have a force that is pulling it to the outside of the circle. And so it won't just, even when the bucket is upside down in that part of the circle, the water will stay in the bucket. Of course, and so that the reason for that is that the acceleration, the bucket has to be accelerating, because in order to keep it going around in that circle, the only way it could not be accelerating is if it was just going straight in a straight line. To keep it going in the circle, you have to continuously accelerate the bucket to keep it changing its direction of motion so that it stays in that circle. And that means that you are, one of the fundamental facts is that acceleration is equivalent to gravity. that in fact you can't even tell. That's the so-called principle of equivalence that Albert Einstein started talking about around 1915. That is this idea that if you're in a completely closed box and it's on the surface of the Earth, you feel gravity in that box, but you could also take that completely closed box and you could accelerate it at 9.8 meters per second, and it would be as if you would not be able to tell the difference between the force associated with that and the force associated with gravity. By the way, in our sort of theory of physics, we can kind of understand how that all works. Let me put that aside for a second. But so you can have sort of simulated gravity by virtue of acceleration, and in particular, you can have it by virtue of circular acceleration that will keep things going in a circle. So the result is that you can make artificial gravity by having a centrifuge where, for example, sort of everybody, if you have a spaceship where the thing is just rotating around at a certain speed, then people can just kind of, if you're just sort of walking around on the exterior, on the inside of that sort of circle that's going around, if it's going around fast enough, then you will be feeling gravity just like gravity on the Earth. And so that's kind of the mechanism. And when people, like in the famous movie 2001: A Space Odyssey that came out 50-something years ago now, the big spaceship that's going to Jupiter in that movie, it has a kind of its main part has a sort of a centrifuge motion, and then people are just sort of walking around it like they would on the surface of the Earth. So that's kind of the way that you achieve artificial gravity in a spacecraft. Now, the question is, how much artificial gravity do you need? I don't think anybody knows the answer to that. People have tried growing all kinds of things. They try growing plants. Sometimes some kinds of things manage to grow in a sensible way without gravity. Other kinds of things get utterly confused by not having gravity. And as I say, there are issues for humans in not having gravity. I think it's a difficult experiment to do, because until one has a spacecraft that has some kind of continuous gravity centrifuge type thing, it's kind of hard to know what's going to happen if somebody's there for a month or something in some reduced amount of gravity, because one just can't do the experiment. I think that this is one of the issues. If we go and make a Mars colony or something, it's living in microgravity, well, not microgravity, in the reduced gravity of Mars. Definitely different things would happen. Given the other, you can kind of work out, if all else was equal, how would a child who grows up on Mars, what would they be a lot taller than a child on Earth? Would they be, what characteristics would they have? We weren't designed for that, so to speak. So I don't think we really know. So that's one of these challenges. Let's see. There's a question here from. There's a question from Mikhail about, can a quantum computer solve the protein folding problem? Okay, let me explain an issue. So I talked about proteins. A protein is this sequence of amino acid units, and some of them are quite short, but some of them might be up to maybe 1 million units long of this protein. And the question is, you've got this long string of amino acids, and each one of those is a clump of, I don't know, a few tens of atoms. And the question is, what will happen to this big string? If you put it in water, for example, is this big string going to just be a big string, or is it going to all curl up and make a single blob? So the question of what it actually does is a difficult question. There are forces that act between these different amino acids, these different residues, they're called, these different lumps of these different amino acid units. They have different forces between them. Those forces are forces between atoms, and there are different kinds of forces between atoms. There are forces, for example, in a water molecule, H2O, the atoms, the two hydrogens and the oxygen, are bound together by covalent bonds. What's happening there is that there are essentially electrons in common. Usually in an atom, there's a nucleus, there are a bunch of electrons. that are outside the nucleus. The nucleus is pretty small compared to the atom, maybe 100,000 times smaller than the actual atom. So it's a little point in the middle, but it has an electric charge that keeps the electrons, attracts the electrons to it, and makes a kind of essentially shells of electrons around the nucleus. Well, if you put atoms close enough to each other, things will happen. So, for example, one of the things that can happen is what's called covalent bonding, where essentially electrons are being shared between atoms. And that process of sharing electrons it has the effect of a force that keeps those atoms together. That's one kind of chemical bonding. There's another big one is ionic bonding, where what will happen is an atom will have for example, lost or gained an electron, and that will give it an electric charge, because then the charge on the nucleus doesn't cancel the charge in the electrons, and then there'll be a electrical force of attraction. And that's what happens, for example, in something like a salt crystal. It's a sodium plus, chlorine minus, and those things are electrically attracted. So it's a different force of attraction. There are more smaller forces of attraction between atoms. One particular one is van der Waals forces. So the way those work, those are forces that happen. It's a little bit of a subtle thing. So let's see. Okay, a couple of things to say. A little bit of mathy stuff to say. The forces, electrical forces, there's an inverse square law. That is, the force between two electrically charged things happens to be the same formula for reasons that we now have some good understanding of, at least in our physics project. It's the force between actually it doesn't need all of the apparatus of that's something you could sort of understand in existing physics. But in any case, the force of attraction between two electrically charged things, or repulsion between electrically charged things, has an inverse square amount. So if things a distance r apart, the force goes down like 1 over r squared. Happens to be the same formula as force due to gravity, except gravity is always attractive, whereas electrical forces can be both attractive and repulsive. Okay, so that's a 1 over r squared force. That's the electrical force. The force that well, let's see. There's a force when there's another force which is basically 1 over r to the 6th or 1 over r to the 7th, depending on various details. That's the so-called van der Waals force. Okay, what is the origin of the van der Waals force? If you have, this is a little tricky. If you have two neutral atoms, atoms that have their electrons in these shells around the nucleus and so on, there's a question: will these two neutral atoms, what effect will they have? Will there be a force of attraction between them or not? How will that work? Well, the thing that happens, and it's a phenomenon of quantum mechanics, is quantum mechanics has this feature that, well, the fundamental feature of quantum mechanics is that whereas in classical mechanics, sort of definite things are always happening, things move in definite trajectories, in quantum mechanics, there's always a wide range of different possible trajectories things can move in, and in a sense, they all happen, but with certain weights that lead you to eventually say there's a certain probability for this to happen, and the most likely thing to have happen is the thing that is like ordinary classical physics, but there are these little fluctuations that are around that classical case, but they're little fluctuations that are different. Okay, so that happens all over the place at the level of quantum mechanics, and it happens in atoms, and it happens to the electrons that are in atoms. They're these little fluctuations in the shape of the electron shells and so on around the atom. And that what happens is, and that there's a way to think about that in terms of what are called vacuum fluctuations, which are essentially just a representation of what's happening of this sort of fluctuation process that happens always in quantum mechanics. But the end result of this is the electrons are kind of getting jiggled around in each atom. Okay, so what happens is the electrons in one atom get jiggled around. That produces a certain distortion of the electric. Usually the atom is just a thing that has, it has no electric charge. But what can happen is there's a little bit of polarization of charge. There's the plus charge, some fluctuation will cause the plus charge to all go to the top, the minus charge to all go to the bottom. That's just a fluctuation. It happens for a very short time, maybe a little more than a trillionth of a second, let's say. You just got this little fluctuation that's causing the electric charge to be separated in that way, even though the total electric charge is still zero, but there's just this fluctuation that causes it to separate a little bit. Okay, so you've now got this other atom over here, and that other atom over there, it turns out that when you have an electric dipole, which is what this separated electric charge is, that electric dipole, the electrical forces produced by an electric dipole will induce an electric dipole in something else in which it is possible to induce that. So this other atom over here also has an electron cloud, electron shells around it. And this dipole that has been temporarily formed by a fluctuation in atom number 1 is also has, that will induce a dipole in atom number 2. Okay, so what? Well, okay, what happens then is that you've got this one dipole over here that's been the result of a fluctuation that induces a dipole over here. And then there's a force of attraction between dipoles that happens. And in a sense, where did the dipoles come from? They came from a fluctuation. But the force of attraction is always a force of attraction. Whether the fluctuation is this way or that way or the other way, it's always a force of attraction. And so that's what leads to these van der Waals forces that are a net force of attraction between atoms. And that force of attraction is usually 1 over r to the 6th. And you can actually work that out in a little bit of a mathy thing. The electrical force associated with a single charge is 1 over r squared. By the time you have these two charges together, a dipole, it has an electrical force of 1 over r cubed. And so what's happening is the 1 over r cubed effect of the dipole is affecting the other atom, which then its dipole is having a 1 over r cubed effect on the first atom. And so the result is a 1 over r to the 6th kind of force of attraction between the atoms. Now, it's a little bit tricky. But I said 1 over r to the 6th or 1 over r to the 7th. It turns out there's a relativistic effect that happens because when you are producing that this fluctuation is acting on this other atom, and then that other atom, it's the fluctuation produced in another atom acts back on the first atom. But the point is the effect of the fluctuation from one atom to another, that effect can only travel at the speed of light. So from one atom to the other, the sort of this, okay, I'm having this dipole fluctuating in this direction. Okay, the signal, the electrical signal that is essentially, it's not quite an electromagnetic wave, it's something like that propagates to the other atom, but that takes a certain time. And the net result of all of that is that you have a slightly different force law between the atoms. But that's a force that happens between neutral atoms, and that's one of the forces that can have an effect. There are also these things called hydrogen bonds, which work a little bit differently to do with shared protons between molecules or between pieces of molecules and so on. But in any case, I'm just trying to explain that in addition to the usual covalent bonding that kind of connects things like water together, that's a very strong force of things from shared electrons and ionic bonding. There are these weaker forms of bonding that can also happen. And it's those weaker forms of bonding that happen in this kind of long string of amino acids that cause amino acids to kind of either want to be together or not want to be together. There's also an effect of, if they're in a solvent like water, there's also an effect of, does it want to have water near it? Does it want to be hydrophilic? Does it want to like water and have water near it? Or does it want to be hydrophobic and try and avoid having water near it? And that's another effect that happens in proteins. But the end result is that this long stringy thing will fold up in some particular form. And the question is, what form does it fold up into? So in a first approximation, it will fold up into the form that minimizes its energy. So it will fold up in a form where it's as tightly bound as it can be, where it's kind of like as if you think about this thing, the analogy of kind of a ball rolling around in a landscape, it will tend to go to the lowest energy, the kind of lowest place, the lowest potential energy place in that particular case. And so similarly, when you're folding up a protein, it will tend to go to the configuration that has the lowest possible energy. Now, two issues. One, can it actually get to that lowest energy state? And two, what is the lowest energy state? Let's talk about, can it get there first? Imagine you have a bunch of coins on a table and you push them all together. They'll form a hexagonal pattern. They always do. You push them hard enough, they'll always form a hexagonal pattern. Do the same thing with spheres. There is a configuration of spheres that is the one that people use to sort of arrange spherical fruit in grocery stores and so on. There is a closest packing of fruit, but of spheres, you just take a bunch of spheres and ram them together, they will not make that closest packing. They'll get stuck, they'll jam. So it's not obvious that just because there's a force just because there is a best minimum energy configuration, most closely packed configuration, it's not obvious you'll get there just by jiggling things around. So in fact, in proteins, it is not uncommon to have these chaperone molecules that have the effect of trying to help proteins to fold in the right way, to have this sort of sequence of foldings so that they'll actually end up in the configuration you want them to end up in. And by the way, the shapes of proteins are absolutely critical to their function. I mean, I was talking earlier about alpha helices in proteins, which are the little tubes that can be used to make sort of transmembrane transport of materials into and out of cells. That's in order to make that little tube, you've got to have this protein, this long string, fold up in this helical pattern to make this tube. And so the shapes of proteins are critical. And that's what, for example, a lot of the way that we operate is that proteins are a certain shape and two proteins will fit together like kind of a lock and key, or they'll fit into, one will have a hole and another one will fit into it. And that's a lot of how we operate. And for example, drugs often target particular shapes in proteins, and they'll stick something into that shape, or they'll make some protein have some effect where it is stimulated by virtue of the thing fitting together according to the shape of the protein. So the shapes of proteins are a very critical thing to know. It's even hard to determine if you're just given a protein. It's like, how can you tell what shape this protein is? You've got a bunch of protein and it's in some little, vial in water or something. And it's like, you've got a bunch of protein here. What shape is it? It's hard to tell. There are a couple of methods. One is to make crystals out of the protein. And once you make a crystal, all those protein molecules have to be aligned in some way. And then you can use X-ray crystallography to try and tease out what the shape of those aligned molecules actually is. It's a tricky business because there are usually many shapes consistent with the actual, the X-ray crystallography image that you get out is something which is a kind of transform of the underlying shapes of the molecules. And so, for example, the big thing that Jim Watson and Francis Crick did in 1953 in the sort of quotes discovery of DNA. It wasn't a discovery of the DNA molecule, that was well known. What was figured out was what the actual shape of the DNA molecule was. And a person called Rosalind Franklin had done X-ray crystallography on DNA, and she had managed to get sort of good X-ray crystallography image is. And the question was, given that X-ray crystallography pattern, what was the actual shape of the molecule that produced that pattern? And what Crick and Watson figured out is, if it's a double helix, then you get that pattern. But so it's often a bit of a puzzle problem what the actual shape of a protein molecule is, and there are other methods based on nuclear magnetic resonance or what's medically called MRI, but for same technique is called nuclear magnetic resonance when it's used in chemistry and things. It's a different the approach is to finding shapes that way. But in any case, back to the question of how the shape gets formed. So the question is, if you were just presented with, okay, we've got this protein, and it's got this sequence of amino acids, tell me what shape it's going to fold up into. So the first question is, well, what's the minimum energy shape? Turns out that's a hard problem to answer in general. And why is it a hard problem to answer? Well, it's an example of a problem that is just computationally difficult to answer. So let me try to give an example of that. let's see, here's the sort of the classic similar type of problem is you're given a bunch of cities around the world and you're asked, find the shortest tour that visits all those cities. And clearly, if you've got 10 cities, there are, let's say, 10 factorial, 10 times 9 times 8, et cetera, ways they're about a little bit more than a billion possible orders of those cities in which you can visit them. So one way to find out what's the shortest tour you can make between those cities is just say, let me just enumerate all billion cases and just see how long each one is and work out what the shortest of those is. Well, that turns out, obviously, that takes, you have to go through a billion possibilities. And if you increase the number of cities, make it 20 cities instead of 10, then you're talking about huge quadrillions of numbers of possibilities. It increases very rapidly. So you want an algorithm, a procedure for figuring out what's the fastest tour, what's the shortest tour that doesn't involve just enumerating all the tours. So the question is, can you find such an algorithm? And the problem is it seems to be the case that there are many kinds of problems where you simply can't find really fast algorithms for solving those problems. So the general problem of kind of how do you arrange things in a minimum energy configuration, given certain rules for how different pieces can interact, that general problem is the buzzword is NP complete. That general problem can be such that, at least so far as we can tell, there is no way to solve that problem with an algorithm that doesn't increase, where it doesn't take lots and lots of computer time, particularly as you make the protein, let's say, bigger and bigger. Okay, so that was the state of things. People for many years had been like, how are we going to figure out how protein folding works? Because it seems to, you seem to have to try all these possibilities, or there isn't a fast way to figure out how the proteins will fold. Okay, so gradually over time, there's this competition that's run every year for who can best predict the folding of proteins. And there have been two basic approaches to predicting the folding of proteins. One is the kind of physics approach, where you're saying, let me just look at this protein and let me just imagine that I have a model of the protein. Let me actually figure out how it would move. You start off with this long string of protein in water, and you say, how will it fold up? What are the physical forces that will cause the protein to move this way or that way? How will it fold? That's approach number one. It's kind of the first principles, use physics to see what will happen. Approach number two is basically see what happened before. There are like 30,000 proteins in us, maybe more. Maybe there are 100,000 proteins where the structures are known. You can just take those as samples and you can say, well, look, I've seen this piece of protein and I know from this example that it folds up in this or that way. So I'm going to use that to predict how this new protein that I'm being shown is going to fold up. So that's kind of approach number two. It's kind of like the same thing happened in things like weather forecasting. You can either actually study the physics of the atmosphere, or you can just say, well, I've seen a configuration of winds and low pressure zones and so on that looked like this before. So that's the way I will infer what it's going to do in the second case. So there's been a long running competition between kind of what can be done with the just follow the examples versus what can be done with the pure physics. In recent times, the just follow the examples method seems to be winning. And in particular, using modern machine learning, it's been possible to do pretty well on predicting the structure of proteins. What's actually happening in that case? I was actually just corresponding with a friend of mine who's one of the people who's been doing this particular research, saying, why does it work? And I think the basic answer is because it turns out there are features of proteins that we humans haven't really ever noticed, but which are the large determiners of the final shapes of the proteins, but which those are things which the machine learning system has picked up. So for example, in an alpha helix, it is known that certain sequences of amino acids will always produce this kind of helical structure. Wherever they occur, those are you'll always get that helical structure. Similarly, certain kinds of molecules, certain kinds of amino acid sequences will give you beta sheets. And people have gradually sort of classified this kind of periodic table of different kinds of features that you can identify in proteins, that we can see, oh, that's a such and such kind of feature in the protein. But what seems to have happened with these machine learning systems is that they've identified some more subtle, more complicated features that, that we humans had never really noticed. We didn't give names to them, but they've sort of noticed these features and realized that those features can be used to make good predictions about what the shapes of the proteins will be. So in any case, that's kind of the, so what's happened is, it seems like there's no fundamental reason why it should be possible to know what shapes proteins fold up into, but it seems like proteins are sort of repeating themselves, and they're tending to not do the arbitrarily difficult computational problem. They're tending instead, with the proteins, at least the ones that we look at, tend to have these features that we can identify that will tell us things about how the folding works. Now, an interesting question is, if you just pick a protein at random, what will it do? And let's say we have 30,000 proteins in us. Do we have random proteins, or are the proteins that we have carefully crafted to be proteins that are going to be good for biology? Okay, so I've wondered about this question for like 35 years, and I've asked many protein experts this question. If you take a random protein, what will it do? How will it fold? That's been a very difficult question to answer. And some people say, oh, it's not going to fold nicely. The proteins that we have in biology are ones that have been sort of selected to fold nicely. And other people say you won't be able to tell the difference. So I know from a number of years ago, I just looked at all the proteins where we do know their structures and asked, are the sequences of amino acids apparently random, or are there regularities? The answer is, at that time, I couldn't find any regularities. Now, obviously, these machine learning systems have effectively found regularities in the sequences, which is what's allowing them to make predictions about what the folding patterns will be. But the thing that was so this question of, you pick a random protein. In other words, in the origin of life, for example, could life have just had random proteins, and then life just discovered, oh, this particular protein is good for this, and we'll arrange with this one, and so on, and use that to build all the things that we have in life? Or did life kind of generate the shapes of proteins that we have? I don't think we know the answer to this yet. But in any case, this question of if you're given a random protein, what will it do? And can we predict what shape it will be? We're getting much better at predicting what shape it will be. I'm not sure how well we do yet at a truly random protein that isn't one that actually occurs in an actual organism, or whether there's even any difference, whether it's just random protein. If you can do a protein in an organism, you can do a protein at random. I'm not sure we know the answer to that yet. But that's right. Okay, well, that was, we've been doing a lot of biology discussion here. This question about quantum computers, let me not get into the whole story of quantum computers. That's a long story about quantum mechanics, and I don't think there will be anything magic that happens with that. But I think that in terms of protein folding, the practical problems that are being solved there are being solved with these kind of machine learning trickery, so to speak, more so than a fundamentally different way of doing computing. Well, let's see. Do I have one here that is quick? A lot of interesting questions, but oh my, none of these are quick, I'm afraid. They're all fascinating, but not quick. I think we have to wrap up here, and we've got a lot of very interesting questions. Okay, here's one here. Maybe I can do this. Okay, there's one from William here. Is being able to get to the lowest energy state, is talking about molecules and proteins, the statement that there's a continuous series of energy states the molecule has, that the transformations from one state to another are smooth enough, and that there's no energy state that's isolated from all others. Yeah, okay, so interesting question. So this has to do with the whole general problem of sort of optimization. How do you get the best result from something? So let me give you some examples. So let's say you're trying to find the lowest point on a landscape. How do you do that? You've got this landscape. Let's say the landscape is just a very, it's a sort of smooth shape, and it has a big hole, and you know, it has a peak and it has a big hole and so on. One very straightforward strategy is to start wherever you start on the landscape and always keep following the gradient descent. That means at every given point you're at, look at which place you would have, which direction you would have to go to most go down. So the thing that is the steepest descent, the thing that is sort of the fastest down the mountain from where you are. And if you follow that gradient descent, that steepest descent curve, for a sufficiently smooth shape, that will successfully lead you to the minimum of that landscape. But if the landscape has lots of sort of craggy pieces and so on, that won't work. You'll follow that gradient down, and you'll just get stuck in this kind of mountain lake or something. It's the local minimum, but it's not the global minimum. It's not the river down in the valley. It's just that mountain lake up on the side of the mountain. So there's sort of a question of what are these kind of landscapes like? When you have, for example, let's imagine instead of being positions on a landscape, the positions correspond to different configurations of a molecule, and the height on the landscape is the energy of that configuration of molecule. So then the question is, as you move around on that landscape, you're like changing the configuration of the molecule. And is it the case that you can do this same gradient descent thing and just smoothly go to the minimum energy configuration of the molecule, or are you going to get stuck where the molecule has this shape and that's the local minimum, but you can't get out to the global minimum? So that's a sort of a fundamental problem of these kinds of things, is what is that energy landscape? What is that kind of, what does it look like? And is it like a craggy mountain, or is it like something very smooth where you can easily get to the minimum by just following that steepest descent line? Okay, so in practice, these things tend to be quite mountainous. We don't know how mountainous. I mean, there's another case, which is it could be like, like sort of the idealized golf course or something, where it's all flat, but there's this little hole, and the goal is, you know, and if the ball doesn't get right in the hole, it's like it's not going to know which way to go. There's no kind of big bowl that's leading the ball always to that hole. Instead, it's like you have to hit the hole or it doesn't work. So, you know, what these energy landscapes are like is what determines how easy it is to, whether you can sort of incrementally solve the problem, or whether what kinds of techniques you use. So one important technique that was invented in the 1980s is a technique called simulated annealing. So the idea is, if you are just always following the down the mountain path, then you'll get stuck in the mountain lake. But if you jiggle things a bit, then the chances are, even if you got stuck in that mountain lake, that the jiggling will cause you to jump out of the mountain lake and kind of look around for more ways down the mountain. So the reason it's called annealing is because when if you have a metal, for example, if you take the metal, it's molten metal, and you let it cool down, there's a question of, will the atoms in the metal arrange themselves in a nice perfect crystal, or will they arrange themselves in different ways? Annealing is you heat the thing up again to melt it again to let the atoms rearrange themselves. And so that's kind of the idea, is you're kind of heating the system up so that things are no longer just following those gradient paths. You're kind of jiggling them around with the hopes that you'll jiggle them out of those local minima, those local mountain lake type things, and get them to the global minimum. So that's kind of the first question is, what is the energy landscape? Is it like a mountainscape? And then what kind of a mountainscape? And then it's can you can you use something like simulated annealing to get out of the local minima? And there's sort of a relationship between the optimal annealing schedule, the optimal amount which you like heat it up a bit, let it, this is conceptually heating it up. It's not actually heating it up. It's just adding randomness to the paths that you're following, different amounts of randomness. And the amount of randomness you add is probably somehow connected to the kind of cragginess of the mountains. Exactly how that relation is made is not super well understood. And exactly what kind of mountain shapes you get for different kinds of problems is not well understood. This is a problem that shows up in machine learning also, because the problem of training a neural network, of saying, I want a neural net that's going to behave in exactly this and this and this way, you have a similar kind of optimization problem. You're tweaking the parameters of the neural net to get it to behave in the way that you want. And that's like trying to find this sort of minimum in the mountain by tweaking parameters of the neural net. And so all these techniques like simulated annealing kinds of things are used as part of the process of doing training for neural nets. All right, we better stop here. So thanks very much for lots of interesting questions. And I think next week is US Thanksgiving, and we won't be doing one of these next week, but I'll see you again the week after that. So thanks very much, and bye for now.